Hello dear students. Today, in continuation with my previous two videos on topic meiosis, I am sharing with you the third part of my video where I shall be covering some other aspects of meiosis related to its occurrence, significance, and its comparative account with mitosis. By now, we all know that what is meiosis and how does it takes place. Now, the question comes, where does it takes place? Meiotic cell divisions take place at different times in different organisms, based on which it's referred to as either gametic meiosis, zygotic meiosis, or sporic meiosis. First of all, let us see what do we understand by the term zygotic meiosis. Zygotic meiosis takes place in haploid multicellular organisms. It takes place in haploid multicellular organisms. All the cells present in such organisms are haploid. Zygote formed after sexual reproduction. All the cells usually are haploid and they form gametes. After the fusion of gametes, that is after fertilization, the zygote formed immediately after its formation, the zygote undergoes meiosis to form 4 haploid daughter cells. This type of meiosis that takes place immediately after the formation of the zygote is referred to as zygotic meiosis, and it can be seen in many lower plants with haplontic type of life cycle. Whereas gametic meiosis takes place in unicellular or multicellular diploid organisms, for example, animals. Here in this case, zygote after its formation does not immediately divide meiotically. It first divides mitotically to produce a multicellular diploid individual or a group of more unicellular diploid cells. Here you can clearly see in this picture that after fertilization, the zygote is undergoing mitotic division rather than meiotic division. It is undergoing mitotic division to give rise to diploid multicellular organism. Then at certain specific reproductive age, this diploid multicellular organism undergoes meiosis for the formation of gametes. Because this type of meiosis is taking place just before the formation of the gametes, this is regarded as gametic meiosis. These gametes eventually fuse during sexual reproduction to again give rise to the zygote. So in these organisms, gametes represent the only haploid stage in their life cycle. Next, coming to sporic type of meiosis, let us see where does it takes place. Sporic meiosis takes place in higher plants where they show a diplobiontic type of life cycle. They have both diploid and haploid stages in their life cycle. Here, the diploid zygote undergoes mitosis again, like we just studied in gametic type of meiosis. Here also the newly formed zygote will undergo mitosis to create a multicellular diploid organism which is regarded as sporophyte. Some of the cells in this sporophyte then produce haploid spores through sporic meiosis. So it is the example of sporic type of meiosis, because in this type meiosis leads to the formation of haploid spores and not gametes. The haploid spores thus produced later on undergo mitosis to give rise to haploid individuals which are called as gametophytes. They can be either um male gametophytes or female gametophytes, and these gametophytes in turn will produce gametes through the process of mitosis. These gametes eventually fuse to give rise to the diploid zygote and thus complete the life cycle of the organism. Now coming to my next slide. As botanists, it's of utmost importance to us to know the major differences between plant cells and animal cells undergoing cellular divisions. So we will revise these already discussed points here. In plant cells, we don't have centrioles. Centrioles are absent in case of plant cells, whereas they are present in case of animal cells. Secondly, asters, which are star-like structures formed due to radiating microtubules from the centrioles, are not present in plant cells. Asters are not present in plant cells. Thus, the spindle formation in case of plant cells is regarded as anastral spindle. However, because asters are present in case of animal cells, the spindle formation here is called as astral type of spindle. And next point is that cell division, that is during cytokinesis phase of a cell division. In case of plant cells, there is the formation of a cell plate. However, as we saw in all of our earlier diagrams, the cell division involves furrowing and cleavage of cytoplasm. These are the comparative pictures showing dividing animal and plant cells. We all know that plant cells are more rigid and have a definite shape as compared to animal cells due to presence of cell walls. Here you can see that centrosomes are absent in case of plant cells. However, they are present in case of animal cells. Spindle fibers in the plant cells are anastral because there is no star-shaped aster seen in these spindle fibers. However, in case of animal cells, the spindle fibers are astral. No cleavage furrow during anaphase and telophase, as you can see here that animal cells are dividing by means of a cleavage furrow, a constriction-like structure appearing between the two daughter cells. However, the cleavage furrow cannot be seen in the plant cells. Plant cells divide rather by formation of a cell plate. This is the diagrammatic representation of reproduction in flowering plants showing how micro and megasporogenesis takes place in microspore and megaspore mother cells undergoing sporic type of meiosis. In flowering plants, sporophytic phase represents the dominant plant body, on which flowers are produced as reproductive organs. Anthers of the flower possess microsporangia, whereas ovules represent the female sporangia called as megasporangia. During sexual reproduction, meiotic division helps to produce haploid male as well as female spores, which eventually produce male and female gametes for sexual fusion. This slide shows TS of a mature lily anther as viewed under high resolution microscope. All the images shown in these videos have been taken by me from the internet. Lily anthers are most commonly used in labs to study meiosis as they are large sized and moreover they constantly keep producing male gametes. Staining procedure is usually carried out with the help of stains like acetocarmine or acetoocene. This is another picture showing TS of lily anther, lily young anther undergoing microsporogenesis as viewed under high resolution microscope. Here are some more images clearly showing the microscopic views of various meiotic stages. Here you can see interphase with diffused and unorganized chromatin material. Prophase with condensed chromosomes after undergoing crossing over as well as terminalization. In metaphase, we can see homologous chromosomes arranged along the metaphasic plate. In anaphase, we can see the movement of homologous chromosomes to opposite poles, and in telophase, the groups of chromosomes have reached to their respective poles. Similarly, here you can see some of the microscopic views of meiosis-2 stages. Prophase 2 is showing the two dyads. Metaphase 2 is with the chromosomes aligned on the equatorial plane of the spindle. Anaphase 2 shows the sister chromatids moving to their opposite poles. And finally, the telophase and cytokinesis showing 4 daughter haploid cells. Further, in the next couple of slides, we shall go through the comparative account of mitosis and meiosis. Now, most of us know that mitosis is a kind of cell division meant for the growth of our bodies, whereas meiosis is meant for the production of either spores or gametes in the sexually reproducing organisms. Earlier in these videos, we have already discussed some differences between these two divisions. So here we will revise them and some more of their differences will be discussed here. We all know that mitosis involves just one single division, and that division is an equational division. However, meiosis involves two divisions. First of the meiotic division is reductional, whereas the second one is equational. Mitotic division is regarded as a homotypic division because the resultant daughter cells are exactly similar in structure as well as ploidy level to their parent cells. However, first meiotic division is heterotypic division and the second meiotic division is a homotypic division. Chromosome number of the daughter cells remains same as the parent cell in case of mitosis, whereas in case of meiosis, chromosome number of the daughter cells is reduced to half as compared to the parent. Prophase of a mitotic division is of very short duration. However, we have studied in these videos that prophase of meiosis 1 is of very long duration and it is divided into 5 sub-stages. No pairing of homologous chromosomes takes place in case of mitotic division, whereas we can see the pairing through the formation of synaptonemal complex leading to the formation of bivalence in case of a meiotic division. No crossing over and no chiasmata formation is there in case of a mitotic division, whereas crossing over between two non-sister chromatids of homologous chromosomes take place in case of meiosis. One more difference which is there in case of mitosis and meiosis is that in case of a mitotic division, during metaphase, centromeres are oriented in one single plane. They divide in the late metaphase. However, in case of meiosis, two centromeres of a bivalent are oriented in two different planes. They do not divide in metaphase 1 or anaphase 1, although they do divide transversely during metaphase 2 or anaphase 2, but they don't divide at all in case of metaphase 1 or anaphase 1. Lastly, telophase in case of mitosis is always followed by cytokinesis. However, in case of meiosis, telophase 1 is not always followed by cytokinesis. Sometimes cytokinesis takes place in the prophase 2 phase of meiosis 2. Now last but not the least, what is the significance of meiosis? Meiosis and sexual reproduction are highly important for evolution. They help in maintaining the chromosome number of sexually reproducing organisms. Meiosis is first of all important because it ensures that all the living organisms produced via sexual reproduction contain correct number of chromosomes. To maintain same number of chromosomes in all the generations, a cell must undergo a heterotypic or reductional division. It restricts the multiplication of chromosome number in different generations. Now, I suppose we all can imagine what will happen if the cells never undergo any reductional division. Secondly, the offsprings of sexually reproducing organisms represent the combination of genetic information from both the parents. Meiosis makes genetic variation possible in three different ways, which are crossing over that takes place during Pachytene stage of prophase 1. It involves a physical exchange of segments or genetic information between paternal and maternal chromosomes. Secondly, due to independent assortment of homologous chromosomes during metaphase and anaphase. And thirdly, due to reduction and fusion of gametes. Thus, meiosis increases the genetic variation at a faster rate than mutation, thus forming a basis for evolution by means of natural selection. In the last part of my video, I would like to share some of the relevant questions related to this topic which are usually asked by the examiners. First of all, these are some of the very short answer type questions. First one is, which is the longest and most complex phase in meiosis? Which division from amongst mitosis and meiosis is responsible for healing of wounds? Or sometimes you are asked to name different sub-stages of prophase 1. Or you are asked to name the stage of prophase 1 where crossing over takes place. Or sometimes it is asked like which stage of meiosis is called as bouquet stage. Another question is centromeres of the chromosomes divide during which stage of meiosis? Or you can be asked to define some terms. For example, define the term chiasmata. What do you understand by the term sporic meiosis? Or sometimes you are asked to arrange some events in the correct order of their occurrence. For example, you are asked to arrange chiasmata formation, crossing over, and terminalization. Similarly, here are some of the short answer type questions. Like you are asked to give a brief account of synaptonemal complex. Or you can be asked to write at least 5 major differences between mitosis and meiosis. Or you can be asked that what is karyokinesis and how is it different from cytokinesis. You can also be asked to explain the process of crossing over. Or you can be asked that why does multicellular organisms require a heterotypic type of division? Or sometimes you are being asked to compare the two stages of metaphase 1 and metaphase 2 of meiosis with each other. And regarding the long answer type questions, usually asked questions are give an illustrated account of meiosis in flowering plants. Or you can be asked to write short notes on meiotic prophase, and as a next part of this question you may be asked to write the significance of meiosis. Or you can also be asked to draw only neat and well-labeled diagrams of various phases of meiosis, and also you can be asked to point out its significance along with this. So these are some of the important questions which are usually asked. So I thought to share these questions with you all, and I hope you have understood this process completely. Thank you for being with me. Have a good day ahead.